soluble strictly? Oh. This is the water soluble? Bilin pigments. Can you tell me what is the reason? Linear and that is not the reason. Linear is not the reason. This is also linear only, na? Because it is very good. Here strictly hydrocarbon chains are there, but in case of here there is no hydrocarbon chain. It is just a linear tetrapyrrole. Correct, na? That's why it is water soluble. Remaining these two are what? Lipid soluble. How can we differentiate between one chlorophyll molecule with the other chlorophyll molecule based on the side chain? If you look into the structure of the chlorophyll A, what is the side chain? Methyl group. Correct, na? And if you look into the chlorophyll B, what is the side chain? Aldehyde group. No point of getting confused over there. All right, wonderful. Okay. What is the ultimate goal of a light reactions? To produce an NADH. Everyone answered. Did we really fulfill the goal of light reactions by using cyclic photophosphorylation or not? No. What we produced only? That is energy equivalent. This is called as reducing equivalent. Together called as assimilatory power. Correct, na? So what is the reason why we have not produced both? Because there is a presence of only one photosystem. In case of a non-cyclic, there are presence of two photosystems. Can any one of you tell me what are the prerequisites required for the production of ATP? Prerequisites means what are required. Can I say? Very good. Proton pump, proton gradient and ATP synthase enzyme. That is a direct question asked in the NEET. Very good. Very, very good. What are LHCs? Can you define me?
Light harvesting complex.
What exactly it means? What is composed of light harvesting complexes? Center. Antenna pigments are accessory pigments. Reaction center also called as principal pigment, exclusively made up of chlorophyll A. Whereas antenna pigments or accessory pigments are made up of
Chlorophyll B,
and carotenoids. Perfect, perfect. Okay. What is the chemiosmotic hypothesis? This is the hypothesis which is explaining how the ATP can be synthesized by proton motive force, which is explained by Peter Mitchell. Very good. Perfect, perfect. That is what we read. Now let's enter into the most difficult concept where really tough questions appear in the exam, where most of the NEET students are unable to answer the questions. That is dark reaction. Tell me, why do we call dark reaction? It is light independent. Whether light is present or absent doesn't matter. What has to be present? Energy. If energy is present, obviously plant is going to fix the carbon into glucose. Correct, na? The most important statement all of you has to remember. This is also called light independent or why do we call carbon fixation? Because ultimate goal of the photosynthesis is what? Fixing the carbon into glucose. So reducing the carbon into glucose. Now slowly think and answer. Is there any process of oxidation occur in the dark reaction? Slowly. Where oxidation occur? Light reaction. Very good. What is oxidation I am talking about? Water molecule is breaking down into oxygen. So oxygen release only happens in the light reaction. Wonderful. But in the dark reaction only there is a reduction of the carbon into glucose. No point of getting confused over there. And we are calling carbon fixation and carbon. See, I am a plant. I am assimilating or I am incorporating the carbon in my body to convert into glucose. Assimilation means utilization or incorporating into the body to convert into a utilizable form. That's what it is called as a carbon assimilation. Now look into the point. See the picture. In the light reactions we have produced ATP and NADPH. By using this ATP and NADPH, plant is going to reduce the carbon into glucose. Correct, na? The very important statement all of you has to remember, whenever plant is reducing the carbon into glucose, it never reduces the carbon directly into glucose. You are getting the point. Before reducing the carbon into glucose, it converts into a triose phosphates. Why are triose phosphates only? There is a production of glucose. Now all of you answer, where is the site of production of ATP and NADPH? Which side it occur? Is it occur on the granule side or stromal side? Obviously on the stromal side. What is the reason? Because NADPH synthesized towards the stroma, ATP synthase enzyme is also located towards the stromal side. So obviously their synthesis occur in the stroma. So whatever the ATP and NADPH that is synthesized in the stroma, that are used by carbon to convert into glucose. Since energy is present in the stroma only, enzymes are also present in the stroma only. So dark reactions occur exclusively in the stroma only. That is the exact reason if they ask you a question. Okay, there are okay, just a minute. Okay, now try to understand here, when we use the word dark reaction, it is happening in the stroma because of the presence of enzymes. What kind of enzymes are present? That is more important. Of course, there is one of the most important principal enzyme, rubisco, which is a, now there is a question, rubisco is present in all the plants or not? It is present in every single plant on this earth. Apart from rubisco, there is a presence of another important enzyme named as pep carboxylase enzyme. Got it? Since these two enzymes which are required for C3, C4 CAM pathways, they are located in the stroma, dark reactions happening in the stroma. Now all of you understand from now onwards, based on the fixation of the carbon, what? Based on the fixation of the carbon or based on the dark reaction, all the plants on this earth broadly categorized into C3, C4 and CAM plants. Based on what? Carbon fixation. So you all know very well, 95 to 97.5% of the plants, 95 to 97.5% of the plants on this earth belongs to C3 pathway. The plants which are growing in the hot and dry climates, I am not using the word desert, which are growing in the hot, dry climate, they follow C4. I can take a best example of every single angiosperms. I can say more than 18,000 different type of angiosperms follows C4 pathway. In simple words, CAM. What is the full form of CAM? What is the full form of CAM? See, CAM represents crassulacean acid metabolism. Okay, crassulacean. Why we use the word crassulacean? Because the plants which belongs to the family named as Crassulaceae family. Best example, all the desert plants, agave, succulents, opentia, euphorbia, all these are all belongs to Crassulaceae family. That's the reason it is called crassulacean acid metabolism. Right. Now, let's try to understand very clearly. In all our previous exams, sometimes they raise the question separately from C3 and C4 pathway. Nowadays, to make it more complicated, always they compare between C3 and C4 or C4 with CAM or all three together, they will raise the question. Now whether it is a C3 pathway, C4 pathway, CAM pathway, what is the ultimate goal? Fixing the carbon into glucose. That is the ultimate goal of what reaction? Dark reaction. Ultimate goal of light reaction? Production of ATP and NADPH. So by using ATP and NADPH, CO2 is getting reduced to glucose. Now this fixing of carbon into glucose never happens in single step. It always takes minimum of 3 steps. One is called fixation, reduction and regeneration. And trust me, even though you read it 10 times, you never understand. Okay, you never recall. But if you understand once, you can answer any difficult question. So always focus on understanding the concept rather than remembering here. So fixing, we are fixing the carbon, first step, into 3-phosphoglycerate. 3-phosphoglycerate is how many carbon? 3 carbon. Correct na? Here what is the statement I have told to all of you? Whenever we are fixing the carbon into glucose, we are fixing the carbon into triose phosphate, then we produce the glucose. So in that particular step, we are fixing the carbon into 3-phosphoglycerate, which is how many carbon? 3 carbon. Since the first stable or intermediate product to produce this 3 carbon, name of the pathway is C3 pathway, since the product is 3 carbon. Now look into the point. Next one, what is called reduction? We are reducing one form to the another form. So in the reduction, see what we are doing, reduction of 3-phosphoglycerate into, very good, in simple words, 3-PGA into G3P. You got the point? So 3-PGA into G3P. G3P is how many carbon?
3 carbons.
3-PCA also 3 carbon. There is no change in the carbon number. It just reduced from one form to the another form. Finally is regeneration. We'll discuss it very elaboratively, then you automatically get an idea. Okay. So good afternoon, please continue. So everyone just try to understand my statements very correctly. It is all about brilliant understanding. Okay, how difficult it may be, you can answer the questions just like that if you have an idea. Tell me, what is our goal? Fixing carbon
Into glucose.
Wonderful. This is the goal. Correct na? Fixing the carbon into glucose. If I want to fix the carbon into glucose, someone has to accept the carbon. Someone has to accept the carbon, then I can fix into glucose. So in case of a C3 pathway, who is going to accept the carbon? The carbon is accepted by a molecule called as RUBP, which means ribulose 1,5-bisphosphate. The name is ribulose 1,5-bisphosphate. Can you count the number? How many carbon it is? 1, 2, 3, 4, 5. Correct na? So ribulose 5-phosphate is 5 carbon, accepting how many carbons from atmosphere? 1 carbon. Correct na? So 5 plus 1?
6 carbons.
And it is highly unstable compound. So to convert into stable compound, it will break down. You are getting the point. So and what is our rule? Our stable product or intermediate product should be how many carbon? 3 carbon. So how many carbons I am having? 6. So I have a 6 rupees in my hand. I want to purchase the chocolate of 3 rupees. How many chocolates I will get? 2. Correct na? Because my intermediate product should be 3 carbon. I am having 6 carbon, so I will get how many molecules of 3-PCA? 2 molecules of 3-PCA. This is crystal clear, no confusion at all. Now all the students, please try to understand. Our goal is to fix the carbon into
Glucose.
in order to produce one glucose molecule, the Calvin cycle has to turn 6 times. To produce, if I want to produce half glucose,
3 times.
cycle has to turn into 3 times. Correct na? No point of getting confused. Okay. So first we will count for half glucose, we will multiply for 1 glucose. So easy it is. Now look into the point. So for 1 round, 1 RUBP is required. For 3 rounds,
3 RUBP.
I am talking about half glucose first. So for half glucose, 3 rounds cycle has to turn. So every round 1 RUBP, so 3 rounds,
3 RUBP.
1 RUBP is 5 carbon. So 3 RUBP are
15 carbons.
15 carbon. 1 RUBP accept 1 carbon. 3 RUBP accept 3 carbon. 15 plus 3? 15 plus 3 ma 18 carbon. So I got 18 carbon. But what is my goal? My intermediate product should be how many carbon? 3 carbon only. So how many 3 carbons I will get?
6.
I have 18 rupees. I want to purchase chocolate of 3 rupees. So total I will get 6. That's all. So to produce half glucose, cycle will turn.
3 times.
So 3 molecules of RUBP combined with 3 molecules of carbon dioxide, 15 plus 3, 18 carbon, giving rise to 6 molecules of 3-PCA. Correct na? This process is called fixation. Correct na? So how many PCA I got now? 6. How many PGA I got? 6. Each PGA is how many carbon?
3 carbon.
Wonderful. Can you see that? 3 carbons with 1 phosphate group. That's why we can call 3 PGA. Now this is fixation. Now after that, what is the next step? Reduction. What's happening in the reduction? 6 molecule of 3 PGA reduced to 6 molecules of G3P. PGA, what is PGA full form?
Phosphoglyceric acid.
G3P, glyceraldehyde 3 phosphate. They just are reducing age, I mean to say, they both can be interconvertible. We can't say interconvertible. This is the reduced form of 3 PGA. Now, G3P is how many carbon? 3 carbon. No change in the carbon number, no change in the phosphate group. But it just reduced from one form to the another form. But friends, reduction never happened that much easily. In order to reduce one molecule of 3 PGA into G3P, we require 1 ATP, 1 NADPH. So how many G3PG are there? 6. So in order to reduce 6 molecules of 3 PGA into 6 molecule of G3P, we require 6 ATP and 6 NADH. Since so plant struggled a lot in the light reactions and produced ATP and NADPH. Now we are spending here to reduce the 3 PGA into G3P. Now tell me, finally how many G3P are there? 6 G3P. Each G3P is how many carbon? 3 carbon. Out of 6 G3P, 1 G3P is coming out. You are getting my point? 1 G3P is coming out. How many G3P are left out? 5. 5 G3P means how many carbon? 5 G3P means 15 carbon. 5 into 3, 15 carbon. Now every student answer, by using 5 G3P or 15 carbons, how many RUBP I can regenerate? How many I can regenerate? Ma, tell me 5 into 3, how many carbons I have? 15 carbon. RUBP is how many carbon? 5 carbon. Then how many I can regenerate? 3. Correct na? I want to purchase a chocolate of 5 rupees. I have 15 rupees. Then how many I will get? 3 I will get. This step is called as regeneration. Whenever we are regenerating the G3P into RUBP, how many RUBP we are producing?
3 RUBP.
For every RUBP we spend 1 ATP. So for 3 RUBP, 3 ATP. Now let's discuss more depth, how the questions can be framed. Okay, the moment you look at the question, you have to answer it in a brilliant way. So first we are discussing to produce half glucose cycle turns
3 times.
for 1 glucose cycle turns 6 times. No confusion at all. Always understand. So first what's happening? Someone has to accept the carbon. So first 3 molecules of RUBP, total number of carbons are?
53.
Correct na? Each RUBP is 5 carbon. 15 carbon accept 3 carbon, it become 18 and giving rise to 6 molecules of 3 PGA. Now each 3 PGA is how many carbon?
3 carbon.
With 1 phosphate group. Okay. Now 6 molecule of 3 PGA reduced to 6 molecule of G3P. So in order to reduce 1 3 PGA into 1 G3P, how many ATP we require?
6
1 for 1 molecule 1 ATP and 1 NADH. So for 6 molecule we require 6 ATP, 6 NADP. Now slowly think and answer. Finally we got 6 molecule of 3 PGA or G3P. Now out of 6 molecule of G3P, how many G3P are coming out?
1 molecule.
Tell me 1 G3P means how many carbon?
3
3 carbon. Glucose is how many carbon?
6.
So can I use the statement by using 1 molecule of G3P I can produce half glucose?
Yes.
Correct na? So which means to produce half glucose cycle has to turn 3 times. Correct na? Now listen. Now what is the question asked in the 2024 NEET paper? How many number of G3P are required to produce 1 glucose? 2. That is the exact question asked in the exam. Correct na? Because 1 molecule of G3P is how many carbon? 3 carbon. We produce half glucose. For 1 glucose?
2.
Okay. You are telling easily. Okay. Now look into this. Out of 6 molecules, 1 is coming out. How many are left over? 5 are left over. Now 5 molecule of G3P regenerated to RUBP. 5 molecule of G3P is how many carbon? 15 carbon. So by using 15 carbon I have regenerated RUB. Correct na? So in order to regenerate 3 molecules of RUBP, for every RUBP we have to spend 180. You understand? Ask me, I'll explain again. Clear? Now slowly think everyone and try to answer the question. Slowly. How many number of carbon dioxide, how many number of trisekundar, how many number of ATP, how many number of NADPH are required to produce 4 molecules of glucose and 2 molecules of sucrose? Tell me, Ayesha, calculate slowly. Calculating, without calculation you can't. Or you can say it normally also. What happened about Nikita Sanjana? She's coming? Don't know, huh? Okay. Huh? Tell me, Kusma. Guess it. 24 for how many? This one, huh? Okay. Tell me. 20, right or wrong? Say it. 24 carbon dioxide. How many ma? Right or wrong? Say it. 48. Okay. Next. 48. Okay. Now let's look into it. See, when they asked for multiple number, it is always idealized to calculate for one glucose. Then you can calculate for multiple. Now tell me, Ayesha, very clearly, for all of you answer, for a half glucose, tell me, Rishita, how many ATP required for half glucose? For half glucose cycle has to done how many times? 3 times. Correct na? How many ATP required for half glucose? How many? Everyone is looking at this 6. What about this 3?
9.
Correct na? You have to consider this also, right? So for half glucose, 9 ATP. Correct na? Half glucose, how many times carbon dioxide is required? 3 times. Correct or not?
Yes.
How many NADH required?
6.
6. Correct or not? 6 NADH. Can we multiply for 1 glucose? Then how much?
12.
ATP how many?
18.
Very good. 18 ATP, 6 carbon dioxide.
And 12.
Wonderful. You have to remember this correctly. Correct na? Now please answer. I have asked for how many glucose? 18 into 4? 72. 6 into 4? 24. 12 into 4? 48. Correct or not? This is for how many glucose?
4.
Now tell me for 2 sucrose. You read no biomolecules in the first year? Tell me. Sucrose is made up of what, Sanjana? Sucrose, very good, tell me. Sucrose is made up of what? Glucose and fructose. Anyway, fructose is going to convert into glucose. So, one sucrose is made up of how many glucose? One sucrose made up of 2 glucoses only, na? How many sucroses I asked?
2 sucrose.
So, 2 sucrose means how many glucoses?
4 glucoses.
Same. Correct or not? Okay, which will be absolutely same. If I ask you for 1 sucrose, what is the answer? Half of it. How much it is 1 sucrose? 1 sucrose means 2 glucoses. So 18 into 2, 36. Correct or not? 6 into 2, 12. 12 into 2, 24. Any confusions? Now, in very simple words, I will tell to all of you the simplest calculation. Always remember this. Question never ever go beyond this level. I want you to read the PPTs, very clearly. Now, can I say like this? Okay. Now, can I say 6 carbon dioxide, 18 ATP and this is for how many? How many glucoses? 1. Half, 3, 9, 6. 2 glucoses, 12, 36, 24. For 1 sucrose, same. Correct na? Based on that you have to calculate. Correct na? So 90% of the time questions will comes from the calculation only. Okay, I have written every single step very briefly and elaboratively. Okay, so tell me, this pathway is also called as Calvin cycle because of it was studied by Melvin Calvin and his colleagues. For his extensive experiments they awarded the Nobel Prize. Now there is a simple question asked in the exam. Calvin cycle occur in every plant on this planet Earth. In other words, I will tell you, C3 pathway commonly occur in C4 and CAM also. See, every plant on this earth, they have to reduce the carbon into glucose. That is possible via C3 pathway or Calvin cycle. So Calvin cycle is common even in C4 plants and CAM plants because this is the only pathway by which carbon can be reduced into glucose. No point of getting confused over there. So what is the first step? Fixation. Correct na? See, fixation also called as? Why? Can you tell me the reason? Because what is the carbon acceptor? To RUBP what I have done? I have added carbon. Correct na? Since I added carbon, we call it as carboxylation. You can call it as fixation also. And who is the acceptor? RUBP is the primary acceptor. Full form of RUBP, ribulose bisphosphate or ribulose 1,5 bisphosphate. Correct na? Now friends, this is so easy. You understand it till here without having any confusion. Now the real confusion comes here. Okay. Quickly. Okay. Now friends, most of the students will neglect in the exam. Finally, what we produced? Half molecule of glucose. Let's consider cycle turn 6 times. Finally, we produced 1 molecule of glucose. This is clear. Now, my question is very clear. Now, plant in the dark reaction produced glucose. Where it occurred? In the leaf. Correct na? Mesophyll cells of the leaf, production of glucose taken place. Now, you know very well how the sugars can be transported in the plant. Very good. Via phloem. Water can be transported via xylem. Now tell me Kusma, you read na, in the phloem glucose will transport or sucrose will transport? Glucose never ever transport in the phloem. Only sucrose can be transported. But what we produced? Glucose. Then from where the sucrose comes from? Now this is the exact question. This was not asked in the NEET. This was asked in the MCET. And this is very qualitative question. Now look into the picture. Whatever the glucose that we produced, that glucose entered into the cytosol, where it is converted into the sucrose. The same glucose entered into the stroma, where it is converted into starch. So they will directly ask a simple question, where is the site of the synthesis of the starch in the cell or in the chloroplast? Where it is happening? Stroma. Whereas sucrose is getting synthesized in the cytosol. Sucrose is what saccharide? Monosaccharide or disaccharide? You forgot all ma. Okay. Anyway, I'll teach biomolecules, don't worry. Monosaccharide. Saccharide means sugar. Mono, single. Example, glucose, because it is a single sugar. Disaccharide. Di means two. Saccharides are sugar. So it is made up of glucose and fructose. So two different monosaccharides. That's why sucrose is said to be disaccharide. Lactose, disaccharide. Lactose made up of glucose and galactose. We read in the lactoperon also. Correct na? And we read in the biomolecules as well. Then any other disaccharide, maltose. Maltose made up of two glucoses. We'll read in the biomolecules. Anyway, here you remember glucose is finally converted into sucrose and glucose is also converted into the starch. What is starch? Polysaccharide. What is polysaccharide? Poly means many, saccharide means sugar. So many sugars, that is many glucoses come close together, they can form starch. No point of getting confused over there, and it is easy to understand. So this is what pathway? C3 pathway. Now the real challenge here, not the challenge, the real problem is here. Look into that. What is the first step here? Fixation. What is happening in the fixation? RUBP accept one carbon and convert it into 3-PCA. Correct na? So some enzyme has to catalyze this reaction. Correct na? There should be an enzyme which combine RUBP with carbon and convert into 3-PCA. What is that enzyme? That enzyme is only called as Rubisco. From where this enzyme is coming from? We already studied in the yesterday class, chloroplast DNA itself code for this particular protein. And now, what is the full form of Rubisco? The full form of the Rubisco, ribulose 1,5-bisphosphate carboxylase and oxygenase. Why did we use the word oxygenase, friends? Because it can fix the carbon also, it can fix the oxygen also. No point of getting confused over there. Okay. So look into this. The name of the enzyme is, where is this? I think. Anyway, remember, the function of the Rubisco is fixing carbon also and fixing oxygen also. Okay. And very important question for the exam point of view. Rubisco always require magnesium ion as their cofactor. Directly we can get the question in the exam. Now friends, now don't confuse, understand the statements very correctly, because I kept lot of information. By that you no need to touch the book and you can read it very directly. Now focus. This is all you have to understand with a good understanding. Rubisco, which means Rubisco is what? Enzyme, right? This is the enzyme. This is how enzyme look like. Enzyme having what site? Active site, where the substrate will come and attaches. Now this is the Rubisco. Enzyme having how many active sites? One. Now for this one active site there are two substrates. What are the substrates? Carbon dioxide is also substrate and oxygen also, because Rubisco can fix carbon also and oxygen also. But you tell me, according to you, Rubisco has to fix carbon or oxygen? Carbon. Because our ultimate goal of the dark reaction, ultimate goal of the plant is to reduce the carbon into glucose. Correct now? We don't want oxygen. But Rubisco having the feature to fix the other than the carbon. Now if the Rubisco fix carbon, there won't be any problem. But if it fix the oxygen, there is a very big problem. Now what is that problem? Let's try to understand very correctly. Now all of you answer. RUBP, how many carbon?
5 carbon.
Very good. Combines with one carbon from atmosphere. What is the enzyme?
Rubisco.
So now Rubisco is fixing what?
CO2.
Carbon dioxide. Then tell me, Aisha, how many intermediates we will get? How many intermediates? My intermediates 5 plus 1 carbon, 6 carbon. Intermediate means next to product. How many we get? 2 only we get, na? What is our rule? Our intermediate should be how many carbon?
3 carbon.
Don't confuse, okay? If you find small doubt also ask me. Never listen with confusions. We are reading C3 pathway. Why C3 we call the stable product should be?
3 carbon.
So what I am asking you, Rubisco fixed the carbon. Who accepted the carbon? So RUBP combined with 1 carbon and we got how many molecules of 3-PCA? 2 molecules. Here no problem, correct na? If the same reaction occurs, Calvin cycle occurs continuously. There won't be any stoppage of Calvin cycle, no problem at all. Now see when the problem occurs. The problem is occurring, see, same RUBP. Now in the atmosphere there is a no carbon dioxide. What is available now? Oxygen is available. Now again, who is the enzyme? Rubisco. What is the specialty of Rubisco? Can fix carbon dioxide and oxygen. Now don't confuse. You answer, all of you answer. When Rubisco combines the RUBP with oxygen, how many intermediates we will get?
1 molecule.
Wonderful. What is that? How many we get? 1 molecule of 3-PCA. 1 molecule of 3-PCA is how many carbon?
3
3 carbon, wonderful. And how many carbons are left out? How many are left out? Ma, RUBP is how many carbon?
2 carbons left out.
Listen, RUBP is how many carbon?
5 carbons.
Is there any carbon dioxide? What is left out? Oxygen. So we have only 5 carbon. I am having only 5 rupees in my hand. I want to purchase the chocolate of 3 rupees. How many chocolates I will get? Only 1 chocolate I will get. Correct na? How much money is left out? 2 rupees. So same way, RUBP is 5 carbon. We don't have any carbon. Then we get how many intermediates? 1. 1 3-PC of how many carbon?
3 carbons.
And how many carbons are left out? 2 carbons. Correct or not? That 2 carbon containing compound called, very good, 2PGA. You are getting the point. Now you tell me, if this occurs, Calvin cycle continue? No. Because if the Calvin cycle wants to be continued, mandatorily we require 2 molecules of 3-PCA. But unfortunately we got only 1 molecule of 3-PCA. Now you know what the plant will do, C3 plant, what they will do? With a greater difficulty, they convert 2-PCA back to 3-PCA. Then only they continue the Calvin cycle. But this is not that much easy process. Let's try to understand. So whenever Rubisco fix the oxygen instead of the carbon dioxide, that process is called as photorespiration. Now, all of you please understand. Now you all people has to answer. Rubisco is the enzyme. It can fix carbon dioxide also. Now you only answer why it fixed oxygen. See, how many active sites are there? 1. So for one active site, 2 substrates are competing. Whichever is more available, that can be fixed. Now when carbon dioxide is less available and when oxygen is more available, that is the question. So when the photorespiration occur, when the light intensity is high and when temperature is high, when stomata got closed, when carbon dioxide concentration is low. Now friends, now you can ask me, sir, what happens when the light intensity is higher and temperature is higher? What happens? The question is, carbon dioxide completely solubilized. You are getting the point. Which cannot be solubilized? Oxygen. Wonderful. So oxygen also solubilized, but compared to carbon dioxide, oxygen solubilized very less. You all read the respiratory system, na? We will read in the human physiology. When hemoglobin combines with what? Oxygen. Correct na? How much oxygen can be combined with hemoglobin? 98.5%. How much oxygen is left out? 1.5. This is dissolved in plasma. Can you observe? Only 1.5% of oxygen only dissolved in the plasma. Remaining all are associated with hemoglobin. But when you see the carbon dioxide, nearly 9% of the carbon dioxide dissolved in the plasma. So which is dissolving more? So which is available more? Oxygen. Same thing happens in the plant also. So when the temperature is higher, in the mesophyll cells of the leaf, carbon dioxide is solubilized, oxygen is available. Then obviously Rubisco has to fix oxygen. But if the Rubisco fix oxygen, what happens? Calvin cycle will stop. So this reaction is called as what? Photorespiration. Thus question is very clear. Photorespiration is highly a wasteful process. Is that true or false statement? Because unnecessarily the reaction is happening. Now photorespiration also called as C2 cycle. Why? Because we produce 1 molecule of 3-PCA plus 1 molecule of 2-PCA. 2-PCA is how many carbon? 2 carbon. That's why name of the cycle is C2 cycle. You are getting the point, okay? So C3, within the C3 cycle only what cycle is happening? C2 cycle is happening. Don't get confused over there. And what kind of question that we expect in the exam if the question is too tough? If the question is too tough, the question comes from here. All of the following are the possible condition for the photorespiration to occur except. So from there the question can occur, okay, or can appear. Now listen. Now what we have to do, tell me now. What we have to do now? Look into the picture once again, now answer all of you. There are 3 organelles, chloroplast, peroxisome and mitochondria. Okay. Now all of you once again look into the point. This is what, friends? RUBP. How many carbon?
5 carbon.
Correct na? Combined with oxygen. Got it? What we got? I got one molecule of
3 PGA.
and I got one molecule of 2 PGA. Correct na? Now if I want to continue the Calvin cycle, how many 3 PGA are required? 2 molecules of 3 PGA, but I got only one molecule of 3 PGA only. Now you know what I am doing. So plants will struggle a lot and finally they convert this 2 PGA back to 3 PGA. Then only Calvin cycle occurs. Otherwise it never takes place. So in order to convert this 2 PGA into 3 PGA, this 2 PGA has to move towards chloroplast, peroxisome and mitochondria. This is the direct question in the exam. Which of the following organelles are involved in the photorespiration? Okay. Now look into the picture. I'll ask you so simple question, you should have good basics. Now 2 PGA molecule, 2 phosphoglycolate. Where it is present? Chloroplast. The 2 PGA within the chloroplast, it lost its phosphate group. Upon losing the phosphate group, 2 phosphoglycolate converted into glycolate. And what's happening now? Glycolate from the chloroplast entered into peroxisome. Now the glycolate within the peroxisome combined with converted into glyoxylate. Now glyoxylate within the peroxisome converted into glycine. Now glycine from the peroxisome entered into mitochondria. Now the glycine converted into serine. Now serine from the mitochondria entered into peroxisome. Serine converted into hydroxypyruvate. Hydroxypyruvate converted into glycerate. Now glycerate entered into, then glycerate upon spending ATP converted into 3 PGA. So finally 2 PGA we converted back to the 3 PGA. This is all wasteful reaction. And moreover, plants struggled a lot to produce the ATP and NADPH. Correct na? Now here unnecessarily it is spending energy. See here, because if I want to convert the glycerate into 3 PGA, I have to spend energy, which is unnecessarily we are spending. That's the reason we called wasteful reaction or wasteful process. There is a simple question which is asked in the exam. All of you answer. Which is the first metabolite that entered into the peroxisome during C2 cycle? Which is the first metabolite that enters into the peroxisome? What is that? Glycolate is the first metabolite that enters. Now slowly think and answer. Which of the following organelle production of optically inactive amino acid takes place? In which of the following cell organelle the production of optically inactive amino acid takes place? What is optically inactive amino acid? Glycine. Correct na? Why we call glycine as optically inactive amino acid? It's very simple. Every amino acid having what? Carbon. Left side amino group, right side carboxyl group, and one hydrogen and one R group. Correct na? It is the simple structure of every single amino acid. When we consider the glycine, which is said to be most simplest amino acid, in the R group it also contains hydrogen. Correct na? So when we are calling an amino acid is optically active, when we call, when, see, very simple, let's consider this is a glass of water. In this there are certain amino acids are there. When I pass a light, when the amino acid rotate the light right side, we call dextrorotatory. Correct na? When they rotate the light left side, levorotatory. Forget about right or left. First of all, amino acid having the ability to rotate the light, that is called optically active. When they rotate the plane polarized light, that ability called as optically active. If an amino acid wants to rotate the light either right hand or left hand, it should have 4 different functional groups. 4 different. Let's consider, I am writing here methyl group. Can I say all 4 are different? amine, carboxyl, hydrogen and methyl. 4 are different. Then it can easily rotate the light. But in the glycine is what happening? Only, so both are same. Hence it cannot rotate. So out of 20 amino acid, the only amino acid which is optically inactive is glycine. So the question C, they are not asking just where the glycine is produced. They are asking in the C2 cycle which of the following organelle optically inactive amino acid is produced. That is glycine. Correct, no? Where the serine is produced? Mitochondria. Serine is also an amino acid. Correct, na? Very simple. Okay, you have to remember in a way. Only you people can answer the given questions. Always focus on remembering rather than reading. Okay. Now answer the question. Anyway, answer also you can see now. The correct sequence of cell organelles during photorespiration. Remember a short form CPM. Chloroplast, peroxisome and mitochondria. Correct, na? Why it is wrong? Why I have kept this? This is wrong, na? What is this? What is the question? The correct sequence of, 1 second, ma. What something wrong happened? The correct sequence of cell organelles during the photorespiration. This is also completely wrong, na?
Yes.
So because the answer will be chloroplast, peroxisome and mitochondria. This option is also absolutely wrong option. It is not at all correct. Okay. Anyway, so let's complete the C4 pathway also, then we can solve many number of previous year questions. So now don't get confused. This is more interesting now. So now the question is super clear. Okay. Please continue. So the question is super clear here. C2 cycle only happens in which plant? C3 plant. Correct or not? Why? What is the reason? Because C3 plant contain rubisco. Rubisco can fix oxygen also, carbon dioxide also. Now there are two more pathways, C4 and CAM pathways. In the C4 pathway and CAM pathway, there won't be any C2 cycle. Then you can ask me why? Because C4 plants and CAM plants, they developed a lot of adaptations. Means adaptation means they have developed a lot of mechanisms by which they can escape C2 cycle. You are getting the point. Understand the concept. Plants that are adapted to the dry tropical regions have the C4 pathway. Now you can ask, sir, how C4 pathway is escaping the C2 cycle? Because of a specialized anatomy present in the leaf called as Kranz anatomy. So the moment you heard the name of a Kranz anatomy, the common statement that you have to recall, it is only present in which plants? C4 plants. Okay. Now friends, don't get confused. This is exactly Kranz anatomy. Suppose if you took a leaf, you do the cross section of a leaf. When you do the cross section of leaf, this is upper epidermis, this is lower epidermis, which contain guard cells followed by stomata. Now there are two cells are there. These cells are said to be mesophyll cells. Okay, and these cells are said to be what cells? Bundle sheath cells. Okay, you try to watch previous class, it become clear. Okay, mesophyll and bundle sheath. You can observe by the picture itself, when you look into the mesophyll cells, all the mesophyll cells are loosely arranged, having lot of intercellular spaces. And most important point, most of the mesophyll cells are located towards the surface. Correct or not? Most mesophyll cells are located very close to the leaf surface. Look into the bundle sheath cells, friends, they are located very deeper. Is there any intercellular spaces that you can found? No, they are very tightly, compactly attached, and they do not have xylem, they are very close to the xylem and phloem. Correct, na? Now what is Kranz anatomy? How does this Kranz anatomy occurs? Kranz anatomy wants to takes place compulsory, we require both mesophyll also and bundle sheath also. Now the direct question, how mesophyll and bundle sheath cells are connected with each other? That is the question. They both are connected with each other via plasmodesmata. What is plasmodesmata? Very good. In simple words, we can call cytoplasmic bridge. This is cytoplasm of one cell, this is cytoplasm of another cell connected with each other by a bridge. That bridge only plasmodesmata. So, two cells connected with each other via plasmodesmata. That is very clear. But what is our goal of the C4 pathway? What is the goal of every plant? Fixing the carbon into glucose. This is ultimate goal. But there is a problem with the C4 plants. They have to avoid which cycle?
C2 cycle.
Which means plant should not fix oxygen. They have to fix only carbon. So how they are making it happen? Very good. Correct, Rathar. Please continue. Now look into the point. You have to understand this extremely depth compared to the C3. It is very tricky. First, I'll give you the overview, then I'll explain you extreme depth. Okay. This is what cell? Mesophyll and bundle sheath. Friends, can you see this? This is plasmodesmata. Okay. Now focus. In case of a C3 pathway, if I want to fix the carbon, someone has to accept the carbon. Who accepted in C3? RUBP. How many carbon?
5
Wonderful. Similarly, in the C4 also, ultimate goal is what? To fix carbon. So someone has to accept the carbon. Who is going to accept the carbon? The carbon acceptor is a PEP. Phosphoenol pyruvate. We read in the glycolysis itself, right? The 9th step of the glycolysis, we get the phosphoenol pyruvate. So phosphoenol pyruvate is how many carbon? 3 carbon plus 1 carbon.
4 carbon.
So the stable or intermediate product should be 4 carbon. Correct na? That 4 carbon containing compound called as oxaloacetic acid. So the first stable or intermediate product is oxaloacetic acid. Now oxaloacetic acid has to enter into which cell? Bundle sheath cell. Unfortunately, oxaloacetic acid do not have any binding sites to enter into bundle sheath. That's the reason in the mesophyll cells only, oxaloacetic acid converted into either malic acid or aspartic acid. You are getting the point? Then finally what is happening here? Malic acid enters into bundle sheath cell. Very good. Malic acid is how many carbon? Oxaloacetic acid is how many carbon?
4 carbon.
Oxaloacetic acid converted into malic acid. That is also a 4 carbon. Now malic acid entered where? Which cells? Bundle sheath cells. Now so far understand very clearly without having a confusion. Now we read one statement that Rubisco is present in every plant, which means Rubisco should be present in C4 plant also. But in C4 plants there are 2 enzymes. Number 1 enzyme, it is called principal enzyme. Principal means major. Pep carboxylase enzyme or pepcase. In the in the bundle sheath cell we have an enzyme called as Rubisco. Okay. Now I'll ask a simple question you all has to answer. So what is the function of pep carboxylase? First PEP accept carbon. Correct na? Converted into? So that production of oxaloacetic acid is mediated by pep carboxylase. Now finally oxaloacetic acid converted into malic acid. Malic acid entered into? Now malic acid is how many carbon?
4 carbon.
Wonderful. Now malic acid in the bundle sheath cell undergoes a decarboxylation. What is called decarboxylation? Removal of carbon. So this malic acid break down into 3 carbon plus 1 carbon. You are getting the point. Who will take this 1 carbon now? Rubisco. Because in the bundle sheath cell what is there?
Rubisco.
So then Rubisco take that carbon, continue the what cycle? Calvin cycle. You are getting how many carbons are left out? 3 carbons are left out. 3 carbons is called as? Very, very good. Pyruvate. Wonderful. Now that pyruvate from the bundle sheath cell where it is entering? And converted back into phosphoenol pyruvate. Correct na? You tell me, is there any C2 cycle occur here?
No.
Any oxygen fixation occur? No, not at all. Now I'll ask you a simple question to all of you. I'll first let me summarize, we'll read extreme depth of this, then okay. Quickly, so first is what happening? Phosphoenol pyruvate accept 1 carbon converted into oxaloacetic acid entered into bundle sheath. Now malic acid break down into, sorry, oxaloacetic acid never enter. It converted into malic acid enter bundle shape. Then what happening in the bundle shape? By enzyme we will read that it break down into 3 carbon plus 1 carbon. 1 carbon takes by Rubisco. Now my question is, Rubisco having the ability to fix carbon also and oxygen. Now my question is, why Rubisco is not fixing oxygen now? Because Rubisco can fix oxygen, right? Now why it is only fixing carbon? Because we read so clearly, bundle sheath cells are located where, friends? Bundle sheath cells are located extremely deeper. Correct na? Whichever is very deeper, oxygen cannot enter. You are getting? Whichever is very deeper, oxygen cannot enter. So what is left over? Carbon dioxide. Then obviously Rubisco only fix carbon. As a result, C2 cycle can be easily escaped. Okay, this is very simple overview. Now let's understand very depth. So they will also ask you a question in the exam. All of the following plants follows the C4 pathway except maize, sugarcane, millet, sorghum. These are the standard reference examples of the C4 pathway. And I told you na, angiosperm. These are all angiosperms only, no monocots. Correct na? More than 18,000 type of angiosperms are exclusively belongs to the C4 pathway only. No point of getting confused over there. It is easy to understand. So friends, C3 cycle proposed by whom? Melvin Calvin. Calvin, correct. Okay. Now the C4 cycle was proposed by Hatch and Slack. That's why this pathway is called as Hatch, Slack and Core-Stack pathway. Three scientists. Okay. They won't ask, but you try to remember. See, before 2022, the question paper was very easy. Sometimes they used to ask the questions directly from name of the scientist. Okay. But nowadays the question paper was completely upgraded. So now let's discuss the same steps more elaboratively. Now you have to answer the questions very correctly. Read this. Mesophyll cells that are closer to the leaf surface lack of Rubisco. Whereas instead of Rubisco, what is the enzyme? What is the specialty of pep carboxyls? It never fix oxygen. It fix only carbon dioxide. Hence no problem. But the fixed carbon dioxide entered into bundle sheath cell having what? Rubisco. But even though Rubisco is there, it cannot fix oxygen. Why? Because oxygen have a poor access to the bundle sheath cells. Simple concept. Now let's discuss the same thing very elaboratively. In C3 pathway we have fixation, reduction, regeneration. Now let's focus in case of C4. All of you understand very correctly. Carboxylation, breakdown, splitting and phosphorylation. Okay, easy. Now you people only try to answer all. First, carboxylation. Who is the carbon acceptor? Very good. Phosphoenol pyruvate. Accepting carbon. Since we are adding the carbon, we called it as by what enzyme? Pepcase, pep carboxylase, and convert into oxaloacetic acid. Crystal clear. Now oxaloacetic acid cannot enter into the bundle shape, so it has to convert into other acids. So that is called breakdown. Oxaloacetic acid, which is a 4 carbon, by using transaminase enzyme converted into aspartate. Same oxaloacetic acid by using malate dehydrogenase converted into malate. Correct na? So sometimes aspartate also can enter, but most commonly malate enter. Now what is the third step? I'll ask you one question only, you have to answer correctly. Now finally malate entered in the which cells? Wonderful. So malate is how many carbon?
4 carbon.
So it is undergoing what process now? Decarboxylation or splitting. 4 carbon break down into 1 carbon plus 3 carbon. So what is this 3 carbon? Pyruvate. Now what happens to this 1 carbon? Now all of you answer very correctly without having confusion. Focus on the picture so correctly. It is taken by Rubisco, then entered into which cycle? C3 or Calvin cycle. Hence we produce 1 glucose. Correct na? 1 glucose. Now tell me, in order to produce 1 glucose, how many ATP we require? We read that previously. How many? 18 ATP. Very, very good. And how many NADH?
12 NADH.
Wonderful. How many carbon dioxide?
6.
This is clear. So C4 also same calculation come. But first listen. So because Rubisco is there, now it continued the Calvin cycle to produce the glucose. So same thing is happening. Now focus all of you. Now last and final step. What is left out, friends?
Pyruvate.
Pyruvate is how many carbon?
3 carbon.
Where the pyruvate is produced in which cells? Bundle cell. Now pyruvate has to go back to? Correct na? Now see friends, pyruvate converted into? Where? Very good, in mesophyll. Pyruvate from the bundle sheath cell it enter into mesophyll. There it is converted into phosphoenol pyruvate. Now how many times cycle will continue? 6 times. There only one glucose we get. Now which means 6 times pyruvate converted into phosphoenol pyruvate. Every time when pyruvate is converted into phosphoenol pyruvate, we use 1 ATP generally. Correct or not? Then how many ATP we use? 6 ATP. Correct na? But here this is the most important line most of the students even doesn't know. There is an enzyme called as dikinase. Pyruvate dikinase. Which means friends, in order to convert one pyruvate into, it uses 2 ATP. So in order to convert 6 pyruvate into 6 how many? 12. So 18 plus 12? 18 plus 12, 30 ATP. So in the C4 we use as 30, 12 NADH. No need to confuse. Look into the picture very correctly. Till this step it is very clear. Malate, how many carbon?
4 carbon.
Undergoes decarboxylation, break down into carbon dioxide. And production of 3 carbon, which is pyruvate. Now this carbon taken by? Then Rubisco start what cycle? C3 cycle. Then we to C3 cycle, how many times it turn? 6 times. Goal is what? To produce 1 glucose. 1 glucose means 6 times. 6 carbon dioxide, 18 ATP and 12 NADH. This is clear. But where is the pyruvate is located? Bundle shape. But it has to go back to, because so far cycle happened only once. How many times it has to happen? 6 times. So pyruvate has to move to the converted into phosphoenol pyruvate. So how many times it has to move? 6 times. Each time it uses 2 ATP. 6 into 2, 12. So to this 18 ATP we use 12 additional ATP. So hence in the, this is also direct question. So in the C4 pathway we uses 30 ATP. Remaining or same. 6 carbon dioxide, 12 NADH. Okay, never get confused with that. Understand, right? Okay. Now don't confuse, okay? Okay. Now, is there any C2 cycle occur in the C4 pathway? No. Any oxygen fixation occur? Obviously not. Where the C4 pathway are generally happening? Mesophyll and bundle shape. C4 plants are generally present in the hot and dry climates. Now, can I conclude the statement? C4 plants are more efficient than C3 in terms of carbon fixation. I am not talking about the number, okay? In terms of fixing the carbon more efficiently, C4 are more dominant. Correct or not? Now, see, C4 plants can perform photosynthesis under adverse conditions like high light intensities and high temperature. Optimum temperature from C4 plants is 30 to 45. But C3 plants here 20 to 25 only. Correct na? See the interesting point. C4 plants can continue the photosynthesis even under low water conditions. Correct na? So I will tell you in the upcoming slide what they are saying. C4 plants continue low water conditions, which is called transpiration ratio. In other words, how many number of water molecules are used by the plant to fix 1 carbon? 1 carbon. If I take C3 plants, friends, in order to fix 1 carbon, it uses 500 water molecule. Which plant? C3 plant. But if you consider C4 plant, in order to fix 1 carbon, it uses only 250 water molecule. If you take a camp plant, desert plants, they use only 50 water molecule. So which is having highest transpiration ratio? C3. So which are water spenders? C3. Water savers? Camp plants. Okay. The C4 pathway are intermediate. Okay. Highest transpiration ratio is always C3. No point of getting confused over there. Okay. Answer the question. Now what's the answer? C4. What is the reason? It is escaping photorespiration, and where it is growing? High temperature. Correct na? Even though it is growing at the high temperature, still it is fixing the carbon. Obviously it is C4 plant only. Okay, no point of confusion there. No answer? Phosphonol pyruvate is the primary carbon dioxide acceptor in C4. C2 plant, there is no carbon acceptor. C3 plants, RUBP. Correct na? So obviously answer is the option A. Okay. Now, correct na? What is sorghum? C4 only. All monocots. So obviously what is that? Oxaloacetic acid. Simple. Answer slowly, slowly. This is a good question. Try it. B. B. B for ball. Okay. Think it properly. Slowly we have to think. Should not be hurry. What is that? Any guess? Right or wrong? Tell. See. What is the question? See here. What is the role of large bundle sheath cells? Correct na? See, we have two cells. This is the upper epidermis, lower epidermis. We have mesophyll. Mesophyll cells having a lot of intracellular spaces. They are located only towards the surface. But in the deeper we are having, where the Calvin cycle is happening? In the bundle sheath only. So, more number of chloroplasts are present only in the bundle sheath cell compared to the mesophyll. So, to increase the number of chloroplasts for the operation of Calvin cycle, and you people answered as the B. Why B is wrong? See here. To enable the plant to tolerate, you only tell me what is the role of bundle sheath cell that helps the plant to overcome the temperature. You tell me, bundle sheath cell, how it will help the plant to tolerate the highest temperature. They do not have any role. What is the role of bundle sheath? Fixing the carbon. What they will do with the temperature. The statement itself irrelevant, inappropriate statement. Correct or not? They do not have any active role helping the plant to tolerate high temperature. Correct or not? Then to protect the vascular tissues from, it is the function of this plant? No. See friends, whether it is a mesophyll cell or bundle sheath cell, they are required for photosynthesis. Now all of you think, yesterday only I explained it you nicely. Which will protect the plant from high light intensity? Which pigments? Carotenoids. We read na? Then carotenoids are responsible for preventing the plant from high intensity of sunlight, such as photooxidative damage. What is the role of bundle sheath here? Correct na? Slowly we have to connect, okay. Presence of mind is very important. To provide this site for, that is wrong na? Yes. Which means what? Bundle sheaths are allowing the C2 cycle to occur. Correct na? That is what the statement says that, which is absolutely wrong statement. See, what you said option B is a kind of partially true, correct na? But that is irrelevant to the given question. And moreover, bundle sheath cell or mesophyll cells do not have any active role in protecting the plant from high temperature. Then you can ask me, sir, how the plant will be tolerated when the temperature is higher. Okay, you will read in the BSc and MSc level, whenever the temperature is higher, plant have lot of defense mechanisms. Okay, you might read this name named as chaperones. Anyone heard? Chaperones are also called as heat shock proteins. They are the proteins help the plant to tolerate with the highest temperature. Okay, I mean, bundle sheath is nothing to do with that. Okay, no need to confuse. Okay, upon reading multiple times you can understand these statements. Okay, answer. Slowly, slowly.
D. D, sir.
D for dog. See, primary carbon dioxide acceptor in C4 plants is phosphoenol pyruvate. Correct na? In C3 plants? RUBP. Now my question, where the C3 pathway is happening? Where bundle sheath cells are there? In C3 plants there are only mesophyll. In the C3, is there any Kranz anatomy?
No.
Wherever Kranz anatomy is there only you get mesophyll and bundle sheath. In C3 plants and CAM plants only mesophyll cells. You understand? We'll see that in the upcoming slide. Whereas, so this is right. Mesophyll cells of C4 plants, what enzyme? Lack of rubisco. Which cells contain rubisco? Bundle sheath. Then mesophyll cells having what enzyme? Pep carbox. So obviously the answer will be the option D. Correct na? Very good. Perfect. The process which makes the major difference between C3 and C4 plants is what? Photorespiration, also called as C2 cycle. Correct na? So clear so far? Okay. So in the next class we will read about the answer this. I believe you answered. Tell me, Sikandar, we read na? Must slowly, must slowly.
2 molecules of 3 carbon compound. Second option.
See, listen, how much intelligent we are whenever we are reading a question, it is idealized to spend 30 to 45 seconds on the given question. Then your anxiety level will reduce, you can understand the given question. What is the question? Activity of rubisco enzyme in photorespiration leads to the formation of. Let's draw it. We already read the same equation. RUBP plus O2, because no carbon dioxide in the presence of rubisco. What do we get? Tell me, Aisha, what do we get? 1 molecule of 3-PZA and 1 molecule of 2-PZA. This is the correct na? Now look into the options. They have given in the first option 1 molecule of 4 carbon compound. So I can eliminate it easily. Option 2 says that 2 molecule of 3 carbon. I can eliminate this. 1 molecule of 3 carbon. They have not given this, that's all. Correct now? If they given, that is also right. And option D says 1 molecule of 6 carbon. Correct? No, slowly, slowly, if you understand, we can answer the given question correctly. Okay. Clear so far? Any doubts? Okay. You are not reading, revise it. Okay. You have a book na? Module. Read the entire C3 cycle by tomorrow, including light reactions. Okay. I'll complete the photosynthesis, then we'll solve 100 questions. Okay. By that, first I'll solve the PYQs again from 2025 to last 15 years. Based on that, we'll solve the practice questions. Then we'll move to the plant growth and development or cellular respiration. Okay. Revise it. Okay. Always remember, revision is the mother of learning. The more number of times you revise, more expert you become. Okay. In science, there is a formula n equal to 6. Okay. Anything, if you read more than 6 times, brain automatically recollects it. Okay. And it is scientifically proved that whatever you read today, if you revise within 24 hours, brain can easily remember up to 15 to 20 days. Okay, so it is idealized to revise. Okay, and when you are reading the biology, always remember in the form of images rather than the text. Any subject, we know, no, if I ask you a question, please think about your mother. What do we think? We think spelling of mother or picture of mother? Picture. Brain always remember information in the form of images, not in the text. So always convert in the form of images and try to remember. Okay, done. Okay. Okay. Okay, tomorrow we will complete this, including this dark camp